from Portugal. My name is Clarisse Nobre. I'm a researcher at the University of Minho at the Center of Biological Engineering. And today, I'm here to talk about the probiotics and the prebiotics in the colorectal cancer prevention. Colorectal cancer is the third most common cancer type, and its development is associated with gut microbiota composition. Gut microbiota holds more than 400 species of bacteria, and most of them are not harmful. Actually, they help us digesting food and protect us against disease and infections. So by modulation of this gut microbiota, by promoting the specific growth of the beneficial bacteria, the so-called probiotics, and keeping a low level of pathogenic species, we can actually improve human health. Probiotics are live microorganisms which, when administered in adequate amounts, they produce beneficial effects to the host. And where do they come from? Initially, they depend on mother's microbiota and on the mode of the liver. And also, they depend on the birth environment. They are rarely dependent on genetic factors, and after infantry, probiotics are supplied from raw foods such as yogurts and cheese, and by probiotic supplements. Probiotics that have been used in colorectal carcinoma therapy are mainly bifidobacterium and lactobacillus. And how do they act? They increase the beneficial bacteria, they bind and degrade carcinogenic compounds, they produce antibacterial compounds, they decrease the activity of harmful bacterial enzymes, and they also produce anti-carcinogenic compounds. They increase the release of short-chain fatty acids, especially butyrates, that inhibit cell proliferation and induce apoptosis in cancer cells. Other way to increase probiotics is by consuming prebiotics. Prebiotics are compounds that act as a food for the probiotics. And in our lab, we have been producing many prebiotics, especially fruit oligosaccharides that are non-digestible sugars, selectively fermented by the beneficial bacteria, and that promotes the growth of the probiotic bacteria. Fruit oligosaccharides are naturally present in more than 36,000 plants, such as chicory roots, garlic, onion, banana, but their content is low and they are season limited. So industrially, fruit oligosaccharides have been being produced by fermentation also. And in our lab, we have been using mostly fungi that produce enzymes that are able to produce these fruit oligosaccharides. We have been testing penicillin, aspergillus, ariobaciillus to produce these fruit oligosaccharides. And what do? They use the sucrose as a substrate, and they transfer the molecules of fructose into sucrose, and they produce these larger DP sugars such as custose, nistose, fructose. But at the same time, they release large amounts of glucose to the fermentative broth that are not prebiotics. At the industrial scale, normally FOS are produced in two steps. In the first fermentation, the microorganisms produce enzymes, and then the enzymes are purified. And in a second fermentation, FOS are actually produced. The production yields are not very high, are around 55 to 60%. So we have been studying strategies to increase these production yields. One way is to use the whole cells of microorganisms, and so in just one fermentation, we produced these fruit oligosaccharides, and we were able to increase the production yields to 64%. Nevertheless, the purity is still the 56% that normally also at industrial scale is achieved. So we have been, and these These saccharides that represents more than 40% of the sugars that are not fructooligosaccharides are glucose, sucrose, and fructose that decrease the prebiotic, caloric, and cariogenic value of the mixtures. So we have been studying several strategies to purify these fructooligosaccharides, such as activated charcoal columns, microbial treatment, cationic resins, simulated moving bed chromatography, and more recently, we integrate the purification step into the fermentation one. So in one fermentation, we add 2 microorganisms, and this second one, this Saccharomyces cerevisiae without the suctucin, is able to consume the glucose without competing by the sucrose and without neutralizing the fructooligosaccharides during the fermentation. And so in just one fermentation, we are able to produce fructooligosaccharides with 93% of purity and with yields of 70%. So to test now and identify these sugars as prebiotics, we have to look first to the chemical structure of the sugars and and also to the in vitro digestion to see if they are resistant to the harsh conditions of the digestion, such as the low pH of the stomach and also to the enzymes. And then we have also to see if they are actually fermented by the intestinal bacteria. Ultimately, clinical trials have to be done to identify the functionality of these prebiotics. But in vivo models have many disadvantages, such as practical issues, ethical restrictions, sampling of the human gastrointestinal tract. So we have been using in our lab mainly in vitro models. They are quick, easy, and cost-effective. We have installed in our lab a dynamic gastrointestinal model that simulates stomach and the 3 zones of the small intestine. And recently we are developing a new colon system. This simulator is able to simulate the 3 zones of the colon. It is also able to simulate the peristaltic movements of the colon. And it simulates the crosstalk interaction between the microbiota and the epithelial cells. And the name is Colus. So to conclude, in vitro and in vivo studies have shown promising effects of probiotics and prebiotics in the treatment of colorectal cancer. And the administration of probiotics before surgery and during chemotherapy have shown reduced side effects with improved gut environment. However, only few clinical studies with small groups have been reported, and dose and strain dependency of probiotics in colorectal cancer have still to be optimized and verified. So it is important and desirable to continue research on the anti-cancerogenic properties of specific probiotic strains and their mechanisms of action, especially during treatment. Thank you very much for your attention. Thank you very much for the invitation, Dr. Reddy, to be present in your conference. I'm very happy that you were all there to hear me. And see you soon.